Hey, all you cool cats and kittens, it's Miss Ellis. We're going to talk a little bit about transcription. So, you've done the pre video and the web quest, and so now I'm just going to give you some more like details and do the thing that I do. So, you can follow along with your power notes. The central dogma of biology states that information flows in one direction from DNA to RNA to proteins. And so you've got 1 represents DNA, 3 is being rewritten into RNA, and then we use that information to create a protein. And this includes 2 processes, transcription and translation. So on the numbers, transcription is going to be the process that rewrites the DNA message into RNA form that can leave the nucleus, and then translation is where we're going to translate that message into a series of amino acids and eventually create a protein. And so RNA is that link between DNA and proteins. So let's review the DNA structure real quick. So you've got a little chart there. So DNA, number of strands, dose. Sugar, deoxyribose, which is why it's called DNA, deoxyribonucleic acid. And nitrogen bases, obviously A's, T's, C's, and G's. Ribonucleic acid, or RNA, is slightly different. It is single-stranded. The sugar is ribose, hence ribonucleic acid, and it also has 4 nitrogen bases, adenine, guanine, cytosine, and uracil. And so when we are creating the RNA, the uracil is going to replace the base thymine. And there are 3 types of RNA. So we have rRNA, which is ribosomal RNA, mRNA, which is messenger RNA, and tRNA, which is transfer RNA. And of course, the names are basically their functions. So messenger RNA, its function is to read the DNA and carry the message from the nucleus to the ribosome. DNA can never, ever, ever leave the nucleus. And we're not changing DNA, we're simply rewriting it into a form that can leave the nucleus and go to the ribosome. So there's just a little animation. So see, DNA, not changed, written into an RNA message that can leave. Transfer RNA, its function is to go pick up specific amino acids according to the codon and transfer them from the cytoplasm over to the ribosome. And it's the ribosome that's going to link the amino acids together into a polypeptide chain. And this is happening in the cytoplasm. Ribosomal RNA is basically just used with proteins to create a ribosome. This happens, they're located in the cytoplasm as well. So I'm going to go through the basic steps of transcription. I don't want you to jot this down. You're just going to kind of watch. So the first thing that happens is we're going to create RNA using the DNA template. And so we're just basically transcribing a section of the DNA, not all of it, just a section that is going to code for a specific protein that's needed in an organism. So step 1, RNA polymerase, ASE, we know that's an enzyme, separates the DNA nucleotide, so it kind of busts open those hydrogen bonds. Then free-floating RNA nucleotides will match up with the DNA using complementary base pairing rules, except U instead of T. The mRNA will then break free and into a separate piece. And then some messenger RNA, some processing takes place. So it's kind of like a rough draft, and it kind of has to be finalized before it can be submitted, so to speak, to go over and actually create a protein. Certain sections aren't going to be used, and so those are called introns. And this is counterintuitive. Introns are taken out, and the portions that are kept are the exons. They're called exons because they are actually expressed. And then the messenger RNA will travel to the ribosome, and then DNA recombines. I can't stress this enough. We aren't changing DNA. We are simply rewriting it in a form that can leave the nucleus. So there's your DNA separation. Now we're transcribing using base pairing rules. Notice that we use U instead of T. Messenger RNA leaves. It's going to go do its job out in the cytoplasm. Oh, and then the DNA recombines. Notice we have not changed the DNA. Okay, so on your notes template, there are some boxes to kind of jot down what's happening. So the first part of transcription is called initiation. I'm abbreviating here. So there are these transcription factors, which of course are proteins, and RNA polymerase. Those combine to create a transcription initiation complex. That's going to bind to a series of A's and T's called a Tatabox promoter. Promoter simply promotes or starts this process. RNA polymerase cannot bind to the promoter without these transcription factors. So it's just this big complex that's going to get this party started, so to speak. Step 2, excuse me, is the elongation portion of transcription. This is where RNA polymerase moves along the DNA. It's going to untwist and unwind the DNA helix and then add RNA nucleotides at the 3' end. So again, notice that we start here, we add nucleotides in this direction, building it in the 5' to 3' direction, adding on to the 3' end every time. As the complex moves down the DNA strand, the helix will reform, okay, because we haven't changed it. We just opened it up for a hot tick to use the template to make a copy. And you can see the RNA straggling away from the DNA template. That's just an enlarged version of the diagram I just showed you so you can see it a little bit easier. And everything, all the prime ends are labeled, and that big pink blob is RNA polymerase. The final step is termination. RNA polymerase will transcribe a terminator sequence. And then at that terminator sequence, the transcription factors are released and RNA polymerase detaches. And then this process is done. Now, like I said, RNA has to be modified, and I'm going to tell you about that in just a couple more slides. So if you're, again, kind of following along with the notes template, there are some additional numbers. These are just identifying structures. So 4 would represent the DNA template strand that we're using to make the messenger RNA. 5 is just kind of showing you a loose RNA nucleotide hanging out, waiting to be added to the messenger RNA. 6 represents the growing messenger RNA strand. And 7 is the gray blob that is RNA polymerase. So I've got a couple animations for you to go through just to kind of show you in real time how this is working. Notice that the big blob RNA polymerase comes in, opens it up. We just open DNA up for a hot tick, and we use that strand to create our messenger RNA, and then DNA goes back together. Okay, here's a second animation. So again, transcription, just the more I can show you, the better. So there's the promoter sequence. You're not going to have to memorize that. You just need to know what a promoter sequence is. Now I got to press this. Okay. So splits open the DNA. We start transcribing the messenger RNA. Again, it's adding to the 3' end. And then we reach the terminator sequence, which just tells everything to stop. So, and then DNA goes back together. So we're not changing DNA. We're just using it to make an RNA. Let me go back to my slides now. So, like I said, the first RNA that's made, messenger RNA that's made, is just basically a rough draft. And we've got some sections that we're not going to use. So this occurs after transcription, but before the messenger RNA leaves the nucleus, and it includes 3 major steps. So step 1, introns are removed and exons are spliced together. So we're actually going to take out the introns, and then we're going to splice all that together and only keep the exons. Exons are kept because they are expressed, and by expressed, I mean they're translated into a protein. The second step is the 5' end gets a little cap. This just helps the messenger RNA bind to the ribosome, and it prevents the strand from breaking down because the cytoplasm, it's rough out there. And so the messenger RNA doesn't last forever. This kind of helps it stay together and not break down too soon. And then finally, what we call a poly A tail is added to the 3' end. It's just a whole bunch of A's, and it just helps messenger RNA exit the nucleus. So cut out the introns, splice the exons, add a cap, add a tail. Now we have our final processed messenger RNA, and it's ready to head out to the ribosome. Eukaryotic RNA is a little more complicated. Enzymes will modify both ends of the pre-messenger RNA. These promote the export of the messenger RNA from the nucleus and to protect it from breaking down or degrading. Like I said, the 5' end gets a cap. It's a modified guanine nucleotide. The 3' end gets a poly A tail, which is about 50 to 250 adenines. Poly mini A adenine, poly A tail. And neither of these are translated into the protein. Like I said, they're just there to help protect the messenger RNA, and they're known as UTRs, or untranslated regions. So they will not code for a protein. They're just there to kind of protect the messenger RNA. Large sections of RNA strands are removed in a process called RNA splicing because those will not be translated. They won't be expressed. And sections of messenger RNA that are spliced out, again, are called introns. Sections of messenger RNA that remain are called exons, and they are spliced together by enzymes called spliceosomes. Spliceosomes. Oh, I've got one more slide. Well, in the words of Michael Scott, I think that pretty much sums it up. Thank you for attending my TED Talk.